Hello, and welcome to this week's episode of the Science Show with Dr. Dada on Jambo Radio Scotland, broadcasting from the city of Glasgow. Today's show will be having a very, very interesting topic, as usual, and it will be this week centered around DNA, nanotechnology, and nanofabrication. So the question of the week, bearing that in mind, is how could DNA, the blueprint of life, transform the future of technology? Well, I'm your host, Dr. Adetunmise Dada, and this is the Science Show on Jambo Radio Scotland, broadcasting live from the city of Glasgow. And don't forget, we are available to be listened to on DAB, and you can also listen and watch us at jamboradio.co.uk. And we invite you to follow the Jambo Radio Scotland channel on YouTube for access to episodes and updates on our show. And this week we have something a little bit special. I've written a few lyrics about the show, and just to start us off, I'm going to give you a taster of these lyrics. If you stick around to the end of the show, you're going to hear the entire thing in a song. But the lyrics go like this. It starts this way. It says, new on the radio, there's a science show where the pros help us learn and grow. Keep us in the know. Tune in and follow us. Listen in as we discuss. Let's unwind, challenge your mind. The Science Talk Show, we're live from Glasgow on Jambo Radio. The Science Talk Show, we're live from Glasgow on Jambo Radio. And the second verse says, bring you the latest news through the interviews we enthuse. We give you expert views, knowledge you can use. From atoms to galaxies, so many mysteries. All the way we learn and play. And so on and so forth. So that's to be continued. So if you listen and wait until the end of the show, you're going to hear that full set of lyrics in song. Great. So today, what are we going to be covering on the show? As usual, we'll be starting with the science news highlights, and then we'll be having an interview with our special guest. And as we said, our guest is a specialist in DNA nanotechnology. So that will be very interesting. And also we're going to have a discussion along the lines of the question for this week. And again, the question for the week is, how could DNA, the blueprint of life, transform the future of nanotechnology? And as usual, we like to have fun on this show. So we'll be concluding with some science jokes. And this week, of course, we're going to have the STEM song, which is the lyrics that I went over earlier in the show. And that's something to look forward to. You won't want to miss that. So stay tuned and don't touch that dial. So let's begin. Let's begin with a quick introduction of our guest. As mentioned earlier, today we have in the studio with us an expert in DNA nanotechnology. His name is Mr. Hamed Ayontola Onilude. We're very happy to have you with us in the studio today, Hamed. How are you doing today?
Yes, I'm fine. Thanks for having me today.
That's great, fantastic. So Hamed is a PhD student, just finishing up his PhD in bioengineering and biomedical engineering. And he is from the College of Science and Technology, the College of Science and Engineering, rather, at the University of Glasgow. And we hope that we can learn a lot more about Hamed during the course of the show. But just one thing, Hamed, of course, your name sounds like a Yoruba name. I'm sure it is a Yoruba name. And I'm Yoruba, so I'm just curious if you'd like to share with us. So your name, Onilude, in Yoruba, as we know, it's a tonal language, and sometimes it can be tricky, right?
Yes, please.
So Onilude, does that mean, because Ilu in Yoruba means a place or a city or a country, right?
Yes.
But Ilu means percussions or drums, right?
Yes, please.
So, that could mean either the percussionist has arrived or the boss of a country or something like that has arrived. So which one is it?
So thank you very much. The second one, actually, when you look at my middle name,
Yes.
which is Anyotola,
Anyotola,
that
indeed.
is Anyon. When someone bears Anyon, that means it's from the drumming family.
Yes, indeed.
So yeah, so like the drum they use back home in Yoruba land, call it the gong gong.
Yeah, like the talking drum.
The talking drum. So
Nice.
it said Anyotola Onilude.
Onilu. So it's
actually
the that's like the
Onilude.
percussionist.
Yes,
That's
please.
awesome.
So it's not actually Onilu. You know,
I
then
see.
they really like things like this in the past. That's part of
Yeah.
our culture. So they
Indeed.
don't want that culture to leave us. So that's one of the reasons why I have Anyotola in my middle name.
Anyotola. Exactly. I guessed as much, but I just wanted to be clear. So that's really interesting because we'll be playing some ilu, some percussions maybe at the end, near the end of the show to some science lyrics. So we'll see how that goes later on. Great. Thanks for sharing that with us.
You're welcome.
Fantastic. So now we're going to start with the science news highlights. So, what do we have on the science news highlights today? We have a few headlines. The first one is about scientific breakthroughs. There's a Forbes article that this is based on, and the title is "Where Are All the Scientific Breakthroughs?" So, what's this about? Well, it says scientific breakthroughs have declined despite advances in AI, nuclear fusion, and mRNA vaccines, states major study. And in spite of notable advances in areas such as artificial intelligence, medicine, and nuclear fusion, major advances in science and technology are becoming more sporadic and less frequent than previous decades, states a comprehensive study published in Nature. This study delved into roughly 45 million scientific papers and 3.9 million patents from 1945 to 2010. And they employed citation networks to gauge whether scientific breakthroughs were reinforcing existing knowledge or creating a significant shift in science and technology. Now, even with significant progress in areas like AI and nuclear fusion, as mentioned before, the researchers concluded that they don't believe such advances contradict their finding of a slowdown in scientific progress. They state that the decrease in disruptive work doesn't necessarily prevent breakthroughs. It simply slows their pace. Now, the detection of things like gravitational waves and development of the mRNA COVID-19 vaccines are highlighted as instances of highly disruptive work taking place at a time when most research tends to be more incremental. The researchers acknowledge the imperfect nature of scientific research with criticisms about funding, the choice of research directions, diversity in research, and the pressures to publish or perish. And they suggest that their findings support a need to rethink how research is conducted, emphasizing the importance of allowing academics the freedom to focus on quality and expand their explorations. Now, this article, if you want to read more about it, is published in Nature, volume 613, from page 138, and that's dated 2023. Great, that's a very interesting story, isn't it? Very, very insightful as well. Good, let's go to the next headline for today then. A pill that mimics insulin could offer new treatment options for people with diabetes, it says. And the headline says, researchers have discovered a molecule mimicking insulin's activity, and this paves the way for potential diabetes oral treatment. So what are the key findings of the research? So one is that they have successfully detailed how a molecule mimicking insulin reproduces insulin's activity in regulating blood glucose levels. And again, the study provides an affirmative answer to a long-standing question about the potential to substitute insulin with other molecules. This discovery opens new possibilities for creating oral insulin substitutes that could replace the daily injections required by type 1 diabetics. Just a little bit more information about type 1 diabetes. Individuals with type 1 diabetes are incapable of producing insulin and need multiple daily insulin injections to maintain their blood glucose levels. But now, this groundbreaking research has confirmed that an alternative molecule can stimulate blood glucose uptake, eliminating the need for insulin. And this study was published in Nature Communications, recently this year. And it's quite interesting what some of the authors have said. One of them, Dr. Kirk, said, scientists have been successful in substituting these mimetic molecules with drugs that can be taken orally. And he continues, although it's still a lengthy journey requiring further investigation, it's exhilarating to acknowledge that our discovery opens up opportunities for oral treatments for type 1 diabetes. So that's quite some sort of good news there, isn't it, in the area of drug discovery. Now, the third highlight that we would like to cover on the show today is about astronomy. It says James Webb telescope finds fewer stars than expected. So what's this about? Well, this is a bit of cosmic news. It turns out that some of the universe's earliest galaxies may have fewer stars than we initially believed. And this is based on observations from the James Webb Space Telescope, JWST. And not only that, but these stars, some of the first ever to light up the universe, seem to be dimmer and bluer than what previous telescopes indicated. So here's the thing, we've been able to capture images of the universe's first galaxies with the Hubble Space Telescope and the JWST and Spitzer Space Telescope, but these were all in what we call the visible spectrum, visible light, or near infrared frequencies. These stars in these galaxies, however, emit light over different wavelengths too, so light of different colors as well. So basically what that means is that we weren't getting the full picture. So the true nature of these stars remained a bit of a mystery as a result. Now, one of the researchers in this new observation, named Popovich, says that we're probably seeing stars when they were just a young 100 million years old or so. But we need to keep in mind that their light has been traveling to us for almost 14 billion years. And one interesting point that Popovich also made is that their measurements show that these galaxies are forming stars at a slower rate than we previously thought. Now, the ball is in the court of astronomers. And as another one of the researchers named Capellari explained, the astronomers will have to work with the data and run it through models to figure out the actual pace of galaxy formation. So that's new, a new result or new knowledge that we're expecting to come out soon in that regard. So plenty of detective work still to be done. If you'd like to read about this, it's published in the Astrophysical Journal Letters, volume 949.2, and that's dated 2023. Great, that's fantastic. So now we're going to get to know our guest a little bit better. And once again, it's nice to have you, Mr. Onilude, with us in the studio.
Thank you.
So to get to know you a little bit better then, would you be able to tell us something about your background? So for example, about your early family life, where you were born, where you grew up, and things like that. We'd like to know that.
All right, thank you very much. Once again, Hamed Ayontala Onilude is my name.
Onilu.
Yes.
I need to get this right?
Yeah, Onilude. At least the drummer boy.
Yes. Excellent.
Yeah, my early life, I grew up in Lagos, where I did most of my early life. From there, you know how Lagos life is actually, it's rough. You have to keep your head up. You have to, if you want to make it fine for you, fine. If you want to do it the other way around, it's fine. But with everything, I think I'm doing fine. After that, I started my career after the university days from Lagos State University. When I finished, started my career in the banking sector, in the financial sector, Access Bank to be precise. So I worked with Access Bank for about 6 years before delving into academics So currently, I'm a staff and a lecturer in Lagos State University. We are lecturing biochemistry. And from there, this is where I am.
That's fantastic.
Yeah.
That's great. It's interesting that you mentioned that you started your career in the banking industry.
Yes, please.
It's interesting. So many scientists do that in Nigeria, don't they?
Yeah.
That's really interesting to know. So, and your undergraduate studies then, what was that in? What did you do in your undergrad?
So, for my undergrad, I did biochemistry throughout in Lagos State University.
I see.
Then when I finished that for my master's degree also, I did biochemistry.
Good.
But now, I won't go further than you. I wanted to bring the questions coming.
No worries. So you mentioned also that you're a lecturer.
Yes.
At LASU.
Lagos State University. Please,
yeah. So what does it take to do that then in LASU? Do you need a PhD to do that, or do you need an MSc, or maybe just a bachelor's is enough to?
Yeah, it depends on the routes. Majorly, before you could start as a lecturer, you need to have a minimum of a master's degree.
Okay.
Or if, depending on your qualification or your, will I say it, qualification when you finish from school as an undergrad, probably you come up with the class of your degree. You know, it could give you an opportunity to start probably as a lecturer with the hope that within certain period you should have your master's degree before you start. You understand? So
that's interesting. That's interesting to know. I think it's maybe very different here, but depending on the institution as well, it can vary, can't it?
Yes. So many things are changing since my own time.
Yes.
A lot of our policies have been put in place, and so many things have changed now. So,
excellent. That's great. Thanks very much for sharing that with us.
Yeah,
of course, we're going to take a music break now, and we'll get to know you a little bit more after the break. So now we're going to listen to a track entitled Kilele. Let's listen to that.
Kana musica, yeah. Coming down with this something. Baby, talk to me. Help me check this thing. I need you desperately. I want you endlessly. Your eyes keep calling me. I feel you constantly. See how you blew me away. I feel in my soul and my heart on a daily. Cricketa, which kind of bonga feeling be this? Kilele, 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 kilele. What it is? Kilele, 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 kilele. Which kind of bonga feeling be this? You are the winner, yeah. And this is getting stronger, hey. So hold me closer, let's make it over. Love you forever, no matter the weather. See how you blew me away. I feel in my heart and my soul on a daily. Rickety, clickata, which kind of bonga feelings be this? Kilele, 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 kilele. What it is? Kilele, 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 kilele. Which kind of bonga feeling? Remix. Me happy, feel better, feel yakata. Sport tag bada, me happy, full ground scatter. Me I go, happy Joe, a new dancer. Come me say, come me down with this something, eh. And there's something in the sweet me, eh. And it is feeling like a movie, eh. And I never been so groovy, eh. Groovy, groovy. Kilele, 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 kilele. What it is? Kilele, 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 kilele. What it is?
Welcome back. This is the Science Show with Dr. Dada on Jambo Radio Scotland, broadcasting from the city of Glasgow. And at this point, we'd like to shout out to our sponsor, the Institute of Physics, IOP Scotland, for sponsoring this episode of the show. And once again, we'd like to remind you that you can listen to us on DAB, and you can also listen at jamboradio.co.uk or through our app. And episodes are also uploaded to the Jambo Radio Scotland YouTube channel. Great. So now we're carrying on in our getting to know Mr. Olunilude, and we're going to really delve a little bit more into the research and the current work that has been carried out by our guest now. So would you like to tell us a little bit more about your PhD studies then? Your PhD research. What's that on?
Excuse me. All right, thank you very much. Currently, I work at the Nanophotonics Research Group, supervised by Professor A.W. Clegg. You understand, that's his group in University of Glasgow, and we currently work do our research at the Advanced Research Center, as is Mazumba Shaw in University of Glasgow. Generally, my research is about reversible immobilization of biomolecules for sensing applications. So that's the very wide area of research. So for my PhD, in the last couple of years, my research has been divided into different sections based on what we want to use that to achieve. The first one, like I said earlier, and you introduced, work on DNA nanotechnology. So the aim of that research is to see how we can reversibly immobilize DNA, which is the genetic makeup of life. That is an example of a biomolecule. You understand? There are other biomolecules too. We have carbohydrates, we have protein, etc.
Yeah.
You know. Now this one is to see how we can reversibly immobilize these DNA molecules on a specialized surface that we have prepared using a specific process known as Florus effect. Now, in the past, most people try as much as possible to immobilize different biomolecules, either to trap them or to conjugate them with other molecules. The essence of that is to see how they can isolate them, you know, and probably characterize them. But there is always a challenge, probably in terms of specificity. That is, if A is going to B, can A and B stay together only without going to C? You know, that specificity. So they've not been able to achieve that. Apart from that, the reusability of that surface is hard, because sometimes when you have a surface you want to use, once the first one is used, it cannot be reused again. So as a result of that, we tend to lose. It's not cost effective. You know, so what this research is bringing to the table is to see how we can now reversibly immobilize this DNA on a surface without losing specificity, integrity, and also making the surface to be reversible. We all know that DNA is the genetic molecule of life. It is actually called deoxyribonucleic acid. You know, there is another one, RNA, which I don't want to delve into now. You know, so. The Florus effect, it's a new field, even though so many people have worked on it before for some times, but we are trying to create another approach to this one. The Florus effect, it's like a tendency that in which perfluorinated compound, I'll break down that. Perfluorinated compounds are like compound that are with a high number of fluorine atoms. You know, what we call fluorine atom is that they are a small molecule with big ego. You know, they have a special characteristic. So we conjugate them with these molecules, you know, and as the number of that fluorine increases on that molecule, their ability to repel other molecule increases, and this is very good for specific interaction in our research. So what we do is to conjugate this DNA with these fluorine molecules and also immobilize them on a surface. So it's a tendency using Florus effect. Florus effect is a tendency that allows this perfluorinated molecule I just explained to react with only perfluorinated molecules You understand? Using non-covalent interaction. Now, it's an advantage for us because it's non-covalent. Most of this research that people have done is covalent interaction in such a way that once these molecules are stuck together, it's hard for them to come up again. So when we use this DNA immobilization, it affords us the opportunity to reversibly immobilize this DNA that has been conjugated with some dyes, and also take them off from the surface with simple solvent wash. Then we can do that several times, put them on the surface, remove them, put them again, remove them, put them again in a repeatable manner. You could imagine the advantage this offers the microarray industries, when you have something that is cost effective. Take for example the current COVID-19 kit that they are using. Imagine if all of us in this building are using just one, whereby you take one, you use it, you clean it with small water, you give it to another person, quickly test it, it's going to save a lot and it's going to help our environment. So for my research, the aim is to see how we can reversibly mobilize this DNA on we call it fluorous pattern surface. That's where nanofabrication comes in, where we pattern that surface and direct that DNA like, hey, DNA, go to this portion. Hey, don't go to portion B, go to portion C, don't go to portion D. So
that's very exciting.
Yeah.
And very well explained as well.
Yeah, thank you. I'm just trying to go a bit to break it down as much as possible so that everyone can understand.
Yeah, no, it's really good. It's really helpful,
Yeah.
and I think you've done a really good job of doing that.
Yeah.
And it's very interesting, isn't it? How a lot of the time, you're looking at something very, very specific, but the end goal is somehow it leads to helping the environment, cutting costs and doing things in a better way. It's that's really, really nice to see. And so now, what stage is your research? Is it would you say close at a high technology readiness level, or is it very much in the works? How would you comment on that?
Yeah, our current research, let's say my research, is a very promising one in the microarray industry and in biosensing application. That's the one I explained earlier was just for the first stage. So having been able to achieve these promising results, we felt that, okay, now can we delve into another one? Since we've been able to achieve this, can we have different DNA molecules of different properties immobilized on the surface? Then can we wash anyone we want out of this immobilized DNA on the surface? When I say immobilized for the science one, let me use the word stick. Different DNAs of different sizes and properties, we put them on the surface, we stick them on the surface.
Yes.
And which one can we direct DNA A away from that surface at our own pace and say, A, we want to wash you away without affecting B? We've been able to do that. So that was what we call multiplexing.
Excellent.
Yeah, we put those DNA on the surface because of our previous achievement in the first immobilization experiment we did. So we're able to also selectively immobilize and remove our preferred DNA from that surface. So which is a very good thing for biosensing application, because like I explained earlier, there are a lot of challenges with different immobilization properties of different surfaces. Now, having done this, we felt, okay, now that we've been able to immobilize biomolecules on the surface, can we also delve further and see if we could use other like metallic nanoparticles? Now the metallic nanoparticles generally have a kind of fascinating optical properties that changes with color upon their interaction with white light. Is made the reason for this because of a phenomenon known as this localized surface plasmon resonance. When nanoparticles, we call them nanoparticles because of their size, that ranges from 1 to 100 nanometer, when they interact with light. So with that, we took advantage of the interaction between gold nanoparticle and special reagents known as we call it tile. We call them tile. You understand? They have sulfur moiety that can easily conjugate with good nanoparticles. Then we took this fluorescence effect further and conjugate the whole moiety of that sulfur-containing tile and the nanoparticles. Those ones will form covalent interaction. Then, after that covalent interaction, we immobilize that complex with the pattern surface that we have created using nanofabrication. Then, unlike what is obtainable in using dyes, when you conjugate dyes with a molecule, sometimes you use fluorescent microscope to view them under the microscope. You understand? It is quite expensive, and you have to change the dye every time you want to use that molecule. In this our current research, what we are doing is to use the visual observation due to the color change of that gold nanoparticles. It's cheap to make. We synthesize them in the lab, and we can easily conjugate it with any molecule we want, immobilize it on the surface, then get the properties via visual observation.
I see. And when you say visual observation, is that something that you can just look at by eye?
Yes, that's what we call it for sensing applications.
Good.
Once this what we call the refractive index on the surface of nanoparticles, once it changes upon their interaction with light or conjugation with other molecules, their color changes. So it's better for us to see something that is colorful. Gold nanoparticles and silver nanoparticles are colorful. So once there is a change in their environment, they change color. So with that, we can know if any molecules of interest is already conjugated with it or not. So we can use that for, and it's cheap. The same thing is reversible, it's specific, it's reusable so many times.
Fantastic.
Thank you.
That's great. That's really great. You'll talk about, thank you very much. Yes, very well deserved applause there. And, talking about, covalent bonds and all these chemistry terms, just takes me back to chemistry class, doesn't it? And you said something about fluorine having a big ego. Would you like to expatiate on that? Tell us a bit more about fluorine's ego.
Yeah, the reason is because when you compare fluorine with other atom in the periodic table, just like when you compare football players, you say, yourself and myself, now let's go to the pitch. Imagine my size to you, you understand? Sorry about that. Anyway,
no.
And let's now imagine I can run faster than you, I can shoot football than you, I can like a keeper, I can catch the ball better than you. Let's say like a ref, I can do the refereeing activity better than you. And now I'm saying, no, as big as you are, this person is very small and you can achieve all these things. And that's why we said fluorine atom, it's a small atom with a big ego. It is very electronegative in nature. You understand?
Great. Thanks for.
So if I say it's electronegative, it can easily attract the electron. It can steal electron from other atoms. So
yes, thanks very much for that.
Yeah,
excellent. So hopefully we learned something there about fluorine's ego. I like storytelling. It's a really nice way to teach, isn't it?
Yes.
Fantastic. So what would you say a day in your life is like? How would you describe, just briefly, like what is a typical day like in your working life?
A typical day, it's like solving problems, and when you want to solve problems, it involves a lot of thinking, a lot of jumping around. Most of the time you see me shuttling from actually working the nanofabrication what's it called, at James Watt nanofabrication place in the University of Glasgow.
Good.
And it's about 5-10 minutes walk to the current Advanced Research Center. So what we do generally is fabricate the surface that we're going to use for onward immobilization. Then you walk down there. Then when you finish with the science in that JWNC, the Nanofabrication Center, then you go to the lab where you do the wet chemistry, prepare those reagents, do whatever you want to do, immobilize. And so a day, it's 9 to 5 of hard work trying to create promising research that can solve scientific challenges and the world in general. Then when you finish from there, you do analysis. What have I got? Is it very good? Is it up to standard? If it's not good, science is not straightforward. Sometimes what you expect may not be what you want to get. You may want A, and the research will tell you, no, I want B, and you want to get A. So many things in a day-to-day activity will dictate the direction of your research. Then from there, you get home and start with the kids, the family. That is our work-life balance.
Good.
Yeah.
Excellent.
Thanks. Yeah.
That sounds like a very, very interesting and exciting life, isn't it?
Yes.
There's science. The nice thing I like about the life of research is that you never know what to expect.
Yes.
And no 2 days are ever the same.
Exactly.
And that's really nice, isn't it? Great. So let's now talk a little bit about the question of the week, which is, how could DNA, the blueprint of life, transform the future of nanotechnology? How could DNA, the blueprint of life, transform the future of nanotechnology? That's a big question, right? In the sense that there's so many, answers or angles you could take in answering that question. But would you like to, in discussing that question, maybe perhaps start, would you like to tell us from the angle of your research? You've kind of touched on it already, but from the angle of your research, how would you say DNA, or the study of DNA, or application of knowledge of how you can manipulate it, could that transform the future of technology? Would you?
All right, thank you. I will start from here. Number one, we say that DNA is the genetic molecule of life. We've always seen it as it helps. That's what keeps us human from generation to generations. So people generally, the science world understands that it's obtainable from our cells. Now, we are not working from the cellular angle. We are working from the in situ angle, So what we are doing is it's going to be on the surface, It can be seen. We can order the DNA, you understand, or isolate DNA from a specific cell, you understand, then put it on the surface. Now, the reason why we are using DNA is because of the self-recognition properties it has, because of the molecular stacking. So we can easily self-assembly that DNA on the surface. So we are not going directly into the cells. Now, the reason, that's the first reason, and the application of that is that it can be used for different sensing application. Now, imagine what you have been able to achieve using DNA only. That, okay, when you have a specific DNA is double-stranded, you have a single-stranded DNA in one part, and you have another single-stranded DNA in one part that's already been conjugated with another molecule. So we can direct each single-stranded DNA to another single-stranded DNA of our choice, and that can be used for several applications, like in gene mutations, and also probably in drug delivery system, you understand, through different conjugation, and all these things are in biological sensing, So I expect this is like 3-in-1 I've just mentioned now. Gene mutation, where you have instead of having gene A in one position, you have gene B there. So you can have a very alarming issue with someone when you have like in sickle cell, you have disease outcome from all these kind of things. Imagine you can easily detect a specific disease or anomaly on time when you have a specific, maybe blood sample, you have it in solution, and you have a fabricated surface, you can easily detect it. So in detection, we can easily use that kind of you can modify that kind of nanofabricated surface. Apart from that, since we've been able to detect that's we can easily see it for biological sensing. That's biological sensing. So with that, we could also use that for drug delivery system, and it can open a lot of areas of science. Like we have there. From there, we can use it for single molecule sensing. That is number one for DNA. We have used it for multiplexing. Imagine you have a lot of molecules around, maybe at a crime scene, and you want to detect just one out of them using this kind of technology. That is the second one, and it's very cheap, easily accessible, repeatable, specific. And the third one, like I said, is we've delved from using the DNA itself. We've now used it. We can conjugate the same DNA with other molecules like nanoparticles that I've talked about earlier, like gold nanoparticle, and with that, any molecule that attaches to that can have a visual color change, which is good for molecular sensing. And apart from that, there are lots of opportunities in that area, which my research group is currently exploring, which I believe you see our our paper from here, our publications.
Yes, indeed.
Thanks.
That's great. Thank you very much for highlighting those things for us.
Yeah.
So yeah, so many applications as you've mentioned, and just one thing I wanted to touch on as well, when it comes to physics, because our sponsor today is actually the Institute of Physics. So
Great.
the question is, where does physics come in when you're talking about DNA manipulation and all these other resulting applications that you've mentioned? So I'll just cover a couple of points on that. So one is the fact that fundamentally, I like to say, and feel free to contradict me on this, that physics is a very fundamental science.
Exactly.
And chemistry is, if you like, based on physics, in the sense that at the fundamental level, the behavioral interaction between molecules, between atoms, is governed by quantum mechanics, which is physics, isn't it?
Yes.
And yes, I know, it's a contentious subject, but
yes,
that's one way to think of it. And really, even in chemistry, you have quantum chemistry, which is really, looking deeper into the quantum physics behind what happens there. So, properties of matter at the nanoscale, understanding quantum mechanics is the most precise way to understand how these things behave. Of course, DNA falls in this realm, if you like. So this is important. So there's a relationship there. There's an overlap there, isn't there? Another thing is the study of thermodynamics. When you're talking about things, you mentioned things like conjugation and sticking, and these are interaction of forces, isn't it?
Yes.
So there's a lot of physics there underneath, behind the scenes. And also there's even a field called biophysics, and the principles of biophysics will help us understand how DNA, for example, interacts with other biomolecules and even with biological entities within a living organism. So do you have something to add to that? Or
yes.
To contradict?
Yeah, I'm not going to contradict for now. It's better I add, because I would shed more light on adding. Now, my research generally, nanofabrication, I'll start from there, has no boundary, especially my research generally, has no boundary as to the field of science.
So it's multidisciplinary.
So yeah, it's a multidisciplinary research, that covers around chemistry, physics, and biology. You understand?
Wonderful.
Even mathematics, is so to say.
Absolutely.
You understand? So like majorly the sensing that we talk about, this with the interaction of our biological molecules with light. You understand?
Yes.
In the visible region or near infrared region.
So that's where the photonics comes in,
yes.
Which is the name of the group.
You understand? So we have other people that are working in those areas. So areas are sectionalized, but we still work. We stay in areas where we normally overlap. You understand? Chemistry, we do a lot of chemistry, a lot of syntheses like gold nanoparticles. Silver general metal nanoparticles. Yeah. Apart from that, if you go to biology in general, DNA, the molecule of life, those three areas cannot be over emphasized. And apart from that, there are other things that come into play. Like I said earlier, for nanofabrication, that can be divided into top-down fabrication or bottom-up fabrication. The top-down I could say is destructive, where you have a bigger molecule, you break it into simpler one of your preference. Then the bottom up, where you synthesize it from the bottom, like atom by atom, you build them together, which is even easier, not destructive, not too energy consuming. All this equipment are through physics, bio related, maybe biophysics, or that's why it's bioengineering. You understand? So physics has a lot of role in our research. As a scientist, you'll have passed through that stage: physics, chemistry, biology. So I love our interdisciplinary. And when you look at the reason why the Advanced Research Center was set up is to enable collaboration, creativity, and also to foster a very good career growth.
Absolutely.
So physics, yes. The answer is big yes. It has a big role.
Fantastic. I'm glad to hear it.
Yeah.
So fantastic. Let's now take another music break, and after that we're going to enjoy some science jokes. So please stay tuned. We're going to listen to a song called Wetam by Tana Musica. We'll be right back.
How you dey? My people, how you dey? Keep it safe. My people, keep it safe. Let's wet your hand with the water. Then apply the soap all over. Scrub all your palm oil, rub over every finger, Under your nails, make you wash 'em. Front of your hands, make you scrub 'em. Back of your hands, make you rub 'em. Then you rinse 'em again. Wetty. Soapy. Rubby. Rinse. Let's wet your hand with the water. Then apply the soap all over. Scrub all your palms, rub over every finger, Under your nails, make you wash 'em. Front of your hands, make you scrub 'em. Back of your hands, make you rub 'em. Then you rinse 'em again. Then rinse 'em again.
All right. That was Wetam by Tana Musica, and this is the Science Show on Jambo Radio Scotland, broadcasting from the city of Glasgow. Again, we'd like to shout out to our sponsor, IOP Scotland, and we'd like to say a big thank you to them for sponsoring this episode of the show. And we'd like to remind you once again that you can listen to us on DAB in Edinburgh and Scotland, and you can also listen and watch us live at jamboradio.co.uk. And of course, episodes are uploaded to the Jambo Radio Scotland YouTube channel. Great. So now we have come to one of the most enjoyable parts of the show, and that's where we share some science jokes. So I don't know if Mr. Onilude has a joke or two to share with us. Otherwise, I can start with one or two. Do you have any science jokes that you know that maybe you think you'd like to share with us? Or should I start?
Please start.
Okay, I'll start in the meantime. So this one, I've selected a couple of jokes that are supposed to be a little bit related to biology and biology related fields. So the first one says, how much room do fungi need to grow? How much room do fungi need to go to grow? It's a joke, so it's a bit of a silly answer, but 3, 2,
how much
1.
room?
How much room? And the key is in room. Well, the answer is yes.
I'm just trying to
go on.
Seriously, I don't know.
Yeah, it's really silly. The answer is as mushroom as possible. I really like this one
because mushroom is a fungi.
So this is a really good one. How much room do fungi need to grow?
You caught me.
As mushroom as possible. It really works, doesn't it? This one.
Good. Yes.
Okay. Good. So there's another one. This one is about cryogenics, which is not okay. I'll just tell the joke. It says, did you hear about the man who got cooled to absolute zero? Do you know this joke?
I know cryogenics is about something below 0 degrees Celsius,
Yeah.
Yeah, sub-zero. So
yeah, something cooled. Yeah, very, very cold. You know, well below 0 degrees and maybe approaching 0 Kelvin, and that's where the clue is. So did you hear about the man who got cooled to absolute zero? Guess what the answer is? It says he's okay now. It was 0, you know, the O is 0.
Yeah.
So that is 0 Kelvin now, right?
273.
Yeah. So
it's fine.
he's okay now, 0 Kelvin. Good.
That's nice.
People who know about the temperature scales will get that one.
Yeah.
Okay, the last one I've got here today is, what did one DNA strand say to the other strand during their date? I thought you'd like this one because, you know, DNA is your thing. What did one DNA strand say to the other DNA strand during their date? Do these genes make me look fat?
Let's be together forever.
Yes. Oh, well, that would work as well. But yeah, well done. But this was the genes, of course, is not, you know, GENS. It's GENES. So genes. So do these genes make me look fat?
That makes sense.
So that's just a bit of a play on words there. Okay. Great. Do you have anyone, or should we go on to the song? No worries.
Now.
Maybe another time. Good. So finally on the show today, we're going to listen to the new song that we talked about at the beginning. So the title of the song is basically about the science show, about this show. So let's listen to it. Let's listen through once, and then we'll go into the lyrics, and then we'll use it as our play out music at the end as well. So let's have a listen.
There's a science show where the pros help us learn and grow, keep us in the know. Tune in and follow us, come join us, we discuss. Let's unwind, challenge your mind. The Science Talk Show. We're live from Glasgow on Jambo Radio. The Science Talk Show. We're live from Glasgow on Jambo Radio. Bring you the latest news through the interviews we enthuse. Give you expert views, knowledge you can use. Atoms to galaxies, so many mysteries. All the way we learn and play. The Science Talk. We're live from Glasgow on Jambo Radio. The Science Talk Show. We're live from Glasgow on Jambo Radio. Talk about the stars up high, planets in our sky, rockets fly, how dinosaurs died, warping space and time. Study the universe, its secrets so vast. You discern, there's much to learn. The Science. We're live from Glasgow on Jambo Radio. The Science Talk Show. We're live from Glasgow on Jambo Radio.
Well, that was the song, and you notice there in the the second verse, we didn't cover that when we looked at the lyrics earlier, so I'll just go over that now. It says, we talk about the stars up high, planets in our sky, rockets fly, how dinosaurs died, warping space and time. Study the universe, its secrets oh so vast. You'll discern there's much to learn. The Science Talk Show, we're live from Glasgow on Jambo Radio. And it says that twice. So that's an interesting song that is really inspired by all the discussions we've been having on this show. And of course, discussions that we hope to have in future. Great. And that's a wrap on this week's exploration into the fascinating world of science. And whether you're an avid science enthusiast wanting to keep up with the latest breakthroughs and impacts on daily life, or you're just eager to learn more about the vibrant scientific activity happening here in Scotland, then remember you're at the right place here on the Science Show on Jambo Radio with Dr. Dada. And of course, we'd love to hear from you as well. So feel free to share your questions, thoughts, and comments about today's show by emailing us through info@jamboradio.co.uk. And don't forget to check out our homepage, www.jamboradio.co.uk/The Science Show for all the latest news and updates on the Science Show. And we would like to extend a big thank you for tuning into this week's edition of the show, and we'd like to encourage you also to keep those radio dials locked, because next time we plan to have another captivating guest on the show. Again, a big thank you to our sponsor IOP Scotland, and a shout out to our wonderful production team, and especially our producer Otuekong Umoren. And again, we'd like to thank Mr. Unilude for joining us on the show today and helping us navigate the world of DNA and technologies that can come as a result of understanding it. And this is Dr. Dada signing off, and until next time, stay curious.
New on the radio, there's a science show where the pros help us learn and grow, keep us in the know. Tune in and follow us, come join us, we discuss. Let's unwind, challenge your mind. The Science Talk Show, we're live from Glasgow on Jambo Radio. The Science Talk Show, we're live from Glasgow on Jambo Radio. Bring you the latest news through the interviews we enthuse, give you expert views, knowledge you can use. Atoms to galaxies, so many mysteries, all the way we learn and play.